A lovely and warm good morning to our most valued and worthy dignitaries, management committee members, sponsor AICTE, respected participants from all over India, and our beloved colleagues. I, Rajaram Das, esteemed Professor Reeves, on behalf of entire organizing committee, welcome you all to the fifth day of Series 2 online FDP program sponsored by AICTE and organized by Reeves. To mark this event a traditional beginning, let us begin with a virtual presentation of lamp lighting and Ganesh Vandana.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
वक्रतुण्डमहाकाय सूर्यकोटिसमप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा
With the blessings of Lord Ganesha, let us start today's event. Dear participants, we are having our first eminent speaker, Dr. Rajnish Kumar Sir from IIT BHU for his session 1 of Day 5 online FDP. Before starting his talk, let me take the opportunity to introduce Dr. Rajnish Kumar to all of you. Dr. Rajnish Kumar Sir is working as an assistant professor in the Department of Pharmaceutical Engineering and Technology at IIT BHU Varanasi. He has obtained his doctoral degree in pharmaceutical science from the University of Punjab University, Chandigarh, India. Sir has completed his PhD research work and was focused on synthesis and study of steroids as 5-alpha reductase inhibitors. Soon after his thesis defense in April 2014, he moved to Karolinska Institute, Stockholm, Sweden, and worked as postdoctoral research associate at the Center for Alzheimer Research, Karolinska Institute, Stockholm, Sweden, until May 2019. In June 2019, Sir accepted a position of assistant professor in the same department. Sir has also been awarded with Japan Society for the Promotion of Science Summer Fellowship. His main interest during his stay in Japan was to explore the target mine data warehouse developed by Miguchi Lab to discover novel drug targets using multi-omics data and artificial intelligence-based tools for pharmacokinetic parameter predictions. His current research is focused on utilization of machine learning tools for novel ADME predictive algorithm for ADME parameters and also for design and development of small molecules targeting a variety of promising drug targets for neurological diseases. With this brief introduction, let us now invite Dr. Rajnish Kumar Sir on board for his eminent talk.
Thank you so much, Mr. Rajaram Das, for generous introduction. I'm really glad and it's my pleasure to discuss and share my research work, which I have been doing for quite a long time. And I thank Professor BVV Ravi Kumar and Professor Viswa Mohan Sahu for inviting me to share the research with you all. I hope you will enjoy the session, and if you have any question during any time, so you can either I came to know that you will not be able to speak or interact directly, but you can write your question in the chat box, and then I'll try to uh, I'll be happy to discuss that actually. So with that, I would like to start with today's topic, that is machine learning based tools for prediction of blood-brain barrier permeation, especially the application of these tools in CNS drug discovery. So I'll be talking about little bit on the different tools which are available, and then a little bit of limitations of those tools and why those tools are important for CNS drug discovery. Okay, so again, little bit more background. So I did my bachelor from Kurukshetra University in 2006, followed by masters from Guru Jambheshwar University, Hisar, and then I did PhD under Professor Manoj Kumar from University Institute of Pharmaceutical Sciences in 2014. And soon after my PhD, I moved to Stockholm, Sweden at Karolinska Institute, and I was working as a postdoctoral researcher. Then I moved to another lab, and followed by that, I got an opportunity to work as assistant professor as well at the same department. And then since 2019 October, I'm working here as assistant professor in the Department of Pharmaceutical Engineering and Technology at IIT BHU. So this is the I think most known place in Karolinska, which is the Nobel Forum. And you know, the Karolinska Institute is the authority to decide Nobel Prize in physiology and medicine. So every year, an assembly of 50 Nobel professors, they sit together and then they discuss to who is the most eligible person to be awarded with Nobel Prize in physiology and medicine. And I was fortunate to witness this ceremony as well, quite a time. So at Center for Alzheimer Research, as the name suggests, the whole division or the whole department was working in the field of Alzheimer research. So it was spread between 3 campuses. We had like South Campus, North Campus, and another campus as well. And you can see that from the pictures on the top, you can see that people in that department were involved in starting from the molecular biology, like structural biology, molecular biology, imaging, and epidemiology. So almost every kind of research is being done there in that department. And as I said, I was fortunate to have this JSPS, Japan Society for Promotion of Science Fellowship, and I spent 2 months in summer of 2019 in Mizuguchi lab with Professor Kenji Mizuguchi, and he's a leading bioinformatician in the field. So, and his interests are in development of artificial intelligence-based tools for drug discovery and ADME modeling. Yeah, so let us come to the topic. Why do we need to predict the blood-brain barrier permeation? So you know that neuroscience is the second largest therapeutic area after cancer, I believe, and then the success rate. But the success rate in neuroscience is only 8% as compared to CVS, that is 20%. And by success rate means here we mean that when you start with the drug discovery, so the drug discovery has different steps actually. We start with we start with target identification, then we come to target validation, then we do hit identification, then we do lead optimization, then we do preclinical assessment, ADME toxicity, and phase 1, phase 2, phase 3 clinical trials. So we have preclinical phase and clinical phase. So starting from, if we start with 100,000 compounds, so in the end we may end up with 1 or 2 compounds which are good in clinical studies. So this lower success rate of CNS drugs as compared to CVS drug is due to this blood-brain barrier actually, because only 2% of CNS drug discovery compounds, they can cross blood-brain barrier. And another problem is that the predictability of animal models for human CNS diseases is low, because every animal species they have their own distinct how to say anatomy, and then they have different for example, one compound that will be permeable, blood brain barrier permeable in rat or mouse, but that will not be in the case of humans. So that variation also happens. So that is also one of the major limitation. So and you know, the drug discovery costs a lot of money. So if we lose any compound in the late phase of drug discovery, so it means that we are losing a lot of money and time on that specific program. So it is always better to filter those compounds, or it is always better to make sure that the compounds which we are developing, so they are blood-brain barrier permeable, and then they can cross the blood-brain barrier and they can show the effectiveness for which they are being tested. So let us come to a little bit of anatomy of blood-brain barrier. So here you can see this is a capillary which is containing RBCs, red blood cells, and this capillary is covered by endothelial cells, tightly packed by endothelial cells, and further these endothelial cells are covered by astrocytes and food or microglia, and then they tightly pack it so that any substance cannot leak out of blood, and only the substances which are essential, for example, glucose, and that is also being transported with the help of some transporters, or oxygen or carbon dioxide, so gases can be exchanged, but not the large molecules. And this is to save the brain from external injuries. Like, you know, we generally we during evolution, the brain has developed its blood-brain barrier to save the brain from external foreign substances. So those might be toxic for the brain. So it protects the brain by making it difficult for substances to get in, and the most substances like nutrients, oxygen, drugs, and proteins are carried into cranium by the bloodstream, but most things can't get from the blood to neurons because of the blood-brain barrier. So from heart, the blood is flown into the arteries, and from arteries it is spread into different organs through capillaries, and then these capillaries they exchanges gases. They have different functions like exchange of gases or exchange of nutrients or other substances. So this is a typical image of a capillary, where you can see that RBCs are lying inside the capillary. Okay, then we will have a little bit on blood-brain barrier transport systems. So you can see that these are the endothelial cells, okay, and then there is a membrane, and then here is the blood, the capillary which is carrying the blood. So there are 5 different mechanisms by which substances are transported from blood to the brain. So the first mechanism is called paracellular aqueous pathway, where you know that the interstitial junction, the junction between the two endothelial cells, through this junction, some water-soluble substances, they can cross this junction, and then they can go into the brain. Another mechanism is transcellular lipophilic pathway, where a substance has to cross the lipid bilayer of this endothelial cell, and then it goes inside the cell, and then it goes outside the cell on the other side of the barrier, so that it can enter the brain. But for this, your substance has to be a lipid soluble, and that should also be having optimal lipid solubility. Otherwise, it will not go into the brain, or it will just, you know, get excreted through the metabolic pathways or excretion pathways. And then we have the third mechanism by which these are some substances are transported, called with the help of transport proteins. For example, we have glucose, amino acid, nucleosides. So these substances, they have, because you know, glucose is a source of energy for brain, and brain is the most brain consumes a lot of glucose because it requires a lot of energy. So then, because glucose it cannot passes through the blood-brain barrier passively, so then we have to have some active mechanisms for bringing it to the brain actually. So in that case, we have specific glucose transporters, and then there are some other transporters which transport some drug substances as well, like vinylalkaloids, cyclosporin A or AZT. And then we have receptor-mediated transcytosis. For example, insulin or transferrin. These substances are transported in they crosses blood-brain barrier through receptor-mediated transcytosis mechanism. And in the end, we have absorptive transcytosis. There are another substances which are generally charged in nature, like albumin or other plasma proteins. So they generally get transported through the blood-brain barrier by using this mechanism. So then we come to the optimal physiochemical properties of CNS drugs. So they, after long research, so people have developed some filters actually, or they made some like generic rules by which they can say that, okay, if the molecular weight of the substance is less than or equal to 450, then it has higher chance to cross the blood-brain barrier. So there are several rules, like hydrogen bond donor should be less than or equal to 3. C log P, that is partition coefficient, that should be less than or equal to 5, because if it goes to more than 5, above 5, then it will be more lipophilic, and more lipophilic means it will be excreted immediately, or it will not be absorbed. And then the hydrogen bond acceptors in that substance, chemical substance, should be less than or equal to 7. The polar surface area should also be less than or equal to 120 angstrom square. If it goes above that, then it might not cross the blood-brain barrier. And the number of rotatable bonds should be less than or equal to 10. So these are some rules which are being used for example, when you do computer-aided drug design. So you filter your ligand library by implementing these rules, and by filtering those compounds with the help of these rules, you make sure that, not sure, but at least you prioritize the compounds which are having more high probability to cross the blood-brain barrier. Okay, so that was about how the blood-brain barrier looks like, and then why it is important, and what are the different physiochemical properties which render our compounds permeable to the blood-brain barrier. So let us discuss a little bit about the AI or machine learning. So AI is it is just a technique where we try to mimic the machine. We try to make machines to do the work which requires some kind of human intelligence actually. So it can be like simple arithmetic calculator, just doing some simple calculations, or it can be a kind of a robot which is doing some maneuvers or some dance like humans actually. So everything comes under the pursuit of AI. So then if we look at this picture here, so you will see that within AI there is a small subset that is called as machine learning, and that machine learning is when the machine or the algorithm learns itself actually, and then within that machine learning circle we can see there is a smaller subset called representational learning, and within that there is a smaller subset called deep learning. So the deep learning is generally using multi-level perceptrons actually, and it is in boom nowadays. So almost every field is using deep learning nowadays. So it's chemistry, biology, genomics, you name it, every field is using deep learning nowadays. So the basic principle of machine learning is using known data. Using known data, you develop a model to predict unknown data. So known data is generally a big enough archive that contains previous observations or previous data, and then you make a model for using that known data, and using different algorithms, machine learning algorithms, and then you predict the unknown data, and that is missing or unseen or not existing, or that is the future data. A very simple example of machine learning application is weather forecasting, for example. So when you just take your mobile and just look at a weather application, so what you will see is it will show you the temperature, or if there is any probability of raining for next 10 days. So that it is forecasting that information, forecasting the temperature, for example, based on temperature of previous days as well as other parameters as well. So those other parameters can be like the position of sun, the speed of wind and direction of wind. So the weather forecasting stations, they collect all those data, and those are called as labeled data. And then by using that labeled data, they make a model using machine learning algorithms. And by that model, they predict or they forecast the weather of next 5 or 10 or 50 days. So that is a very typical example of machine learning application actually. And the similar technique is used in cheminformatics as well, or in ADME prediction modeling, like blood-brain barrier permeability prediction. So another very simple example is, for example, if it is after 2 there is 4, it's kind of time series, we can say 2, 4, 6, 8, and then what will be the next number. So instantly everyone will say that the next number will be 10, because that we know that every time we are adding 2 into that digit to that number. So we add 2 into this number, we get 4, and so on, and then finally we will be having 10 here. So that is simple, and that we can solve because we know the basic rules. Like we can see that we can identify that trend and pattern, and then we can tell what will be the next number, and with almost 100% confidence we can tell. But what about this actually? So can we predict? Can we tell by just looking at the structure which of these compound will cross the blood-brain barrier? If you're able to write your comments in the chat box, I will be happy to see such comments actually. So, but by just looking at the structure, it is quite difficult to say that which of the substances will cross the blood-brain barrier, because the problem is that we do not know the basic rules behind it. Since in this case, we will be knowing that, okay, if we just add 2 into, when we add 2 into this number, so we got the next number. So then we will add 2 into this number, we got this number, and then finally this number can be predicted by just adding 2 into that, because we know the trend. But in this case, it is a little bit difficult for us to just look at the structure and know the trend. So in that case, what we do is we just convert these structures into some kind of descriptors, and those descriptors are the just numbers actually, and then the computer, the machines, or the algorithms can find a trend in those numbers. So they can identify that trend, and they can use that trend to predict the blood-brain barrier permeability of the next compounds, or the test compounds, I would say. So if you look at the AI anatomy, so there are, for example, rule-based system. These are very simple rule-based system. Like I showed you one example of predicting what will be after 2, 4, 6, and 8. So you have input, and you have some hand-designed programs which identify those rules, and then it gives output. And then if we look at the classical machine learning, so we have input and hand-design features, and then we map we do mapping from those features, and we get the output in the form of prediction. So the representational learning, which is nowadays in boom, and especially deep learning. So what they do is they just have input, and then they generate features themselves actually. So now you can see that these features are developed by us, for example, but these features, algorithms are developing those features or reading those features actually. And then they do mapping from features as well as additional layers of more abstract features, and thus mapping from features, and then it gives output. So these are kind of self-learning algorithms. Like deep learning is a self-learning algorithms which learns itself from the data which is being given to it. So then basically ML can be classified into 3 branches: reinforcement learning, supervised learning, and unsupervised learning. And unsupervised learning is further can be classified into clustering, like where we use k-means, mean shift, or k-medoid clustering methods. or it can be dimensionality reduction, for example, PCA, principal component analysis, feature selection, and linear discriminant analysis. While supervised learning can be classified into regression or classification, where in regression we have different methods like decision tree, linear regression, logistic regression, and in classification we have naive-based classification, support vector machines, and k-learner stable classification. So in unsupervised learning, we are not providing the labeled data to the algorithm. So a very simple example of unsupervised learning is, for example, classification of like images or clustering, for example. So when we have a data set of, for example, 100 compounds, and if we want to cluster those 100 compounds according to their similarity with each other, so what we do is we simply calculate the Tanimoto similarity coefficient for each other, for each compound compared to the other compound, and then with the help of that Tanimoto similarity coefficient, we cluster we club those compounds which are having kind of similarity in them based on that Tanimoto similarity coefficient. So that will be kind of unsupervised learning, because we are not telling any of the algorithm that this is this actually. So there is no labeled data in that. While in supervised learning we provide labeled data. We label that data, and then we do a simple classification. Can be by using supervised learning. A simple classification can be like cat and dog picture model, where we are classifying whether this picture is of cat or dog. So what we do is we have to provide several pictures, hundreds or thousands of pictures of cat and dogs, and then the algorithm will learn different features, and then when the model is built, and then we will supply unlabeled images. We will not tell the algorithm that these pictures are of cats or dogs, but the algorithm itself has to tell actually. So that will be kind of supervised learning, where we are supplying the labeled data. Okay, so in classification we just get a binary output, 0 or 1, like yes or no, whether it is a cat or dog, or whether it is blood-brain barrier permeable or not. But in regression model, we get the coefficient of regression. We get a number, that we get a probability number that this substance can cross blood-brain barrier with 0.01% probability or something like that. Okay. So in supervised learning, making a model using training set and let machine use that model to predict test set. So we generally use naive based classifier, where we calculate the probability of event A or B. So that can be calculated by probability of B or A multiplied by P probability of A divided by probability of B. And then we use perceptrons. These are linear separators. So in this case, for example, if these are like red and blue dots. So if we want to classify these red and blue dots, so which line will be best suitable? So which of the linear separator is optimal? And that we calculate with classification margin actually. So that will be least square method, for example. So we calculate the distance of these data points to this line, and then we use square of this distance, and the line which is having least value of this square of different summation of square of different distances, that will be the best fit line. And then we have non-linear support vector machines. We use feature space. So for example, principal component analysis, we generally do classification of like red and blue dot. We can classify using in 3D space actually. So then we have unsupervised learning. Unsupervised learning is training of machine using information that is neither classified nor labeled, and allowing the algorithm to act on that information without any guidance. And generally it is used for classification algorithm or clustering. And then we come to the ANN, that is artificial neural network, which is the basic of deep learning actually. So our human brain contains neurons, and then in billions of neurons we are having. So a single neuron will have kind of cell body, axon, and dendrites. So by the computer scientists, so what they thought is they thought to mimic the human neuron to mimic the way of processing information by human neuron. So they developed this computational algorithm which acts in a similar way. So in this case, we have different inputs, and then we have calculations going in between, and then we have different outputs like that. So there are different kind of neurons, and a single neuron is called as perceptron P. Then you can see that in this mostly complete chart of neural nets, so you can see different arrangement of neurons, like feed forward, where the information is only going in the forward direction. Okay, and then we have radial basis networks, deep feed forward, recurrent neural net, long short-term memory potentials, gated recurrent units, sparse autoencoders, denoising autoencoders, variational autoencoders. So these are perceptrons based on perceptrons, and these are based on recurrent neural nets, and these are based on autoencoders actually. And then we have many more. So there are different deep learning algorithms, deep learning networks, which are being used for a lot of kind of cheminformatics, pharmacoinformatics, drug discovery, and other applications. Okay, so then we come to different open source web servers for blood-brain barrier permeability prediction, and that is the most important part, I think, because this is the application part where you can use these tools for predicting blood-brain barrier permeability of your compounds if you are working in drug discovery field or in CNS drug discovery case. So the first one is blood-brain barrier predictor, where this is based on a script generated automatically by Tree2C program, and then you just enter your molecule in text format, or that is a SMILE format, and then you predict. So you when you enter your molecule structure in SMILE format and you press predict, so you will get the prediction of your compound. And another one is CB ligand blood-brain barrier prediction program. So this one is based on support vector machines and ADA-boost algorithms. So it uses two different algorithms, and then it uses fingerprints, MAX, OpenBabel, MolePrint2D, and PubChem. So all these methods, what they are doing is they have developed a kind of model on the basis of by calculating descriptors of different label compounds. So they collected curated data set from public literature, and then they converted those molecular information into descriptors, either MAX fingerprints or OpenBabel fingerprints, and those descriptors, then they build a model using support vector machines or ADA-boost method, and then that model is showing good predictability actually. So it can predict blood-brain barrier permeability with the high prediction, high accuracy, and so then they develop this web server which can be used for predicting blood-brain barrier permeability of a new compound. So then there is another one that is called ADMET-SAR. So ADMET-SAR is it is again depending upon it includes like several different parameters together with blood-brain barrier permeability as well. Then we have light BBB that is recently developed blood-brain barrier permeability tool. So you can see that it can be used for predicting blood-brain barrier permeability of a new compound, and it takes input as SMILES, so you can use SMILES as input files, and then you can predict the BBB permeability of your compounds. And then there is another one called ADMET-Lab. So again, you see that this BBB permeability model, it has used again support vector machines as an algorithm, a machine learning algorithm, and it has an accuracy of 0.926. That means that it can predict you can predict the compounds, like for example 96 compounds correctly out of 100 compounds. So if you predict 100 compounds by using this tool, so 96 compounds will be predict it correctly. So that is its accuracy for the test set as well. You can see here it's like 0.962. So it is again working the similar way. Either you can input your molecules as a smiles here, and then you calculate the BB permeability, or you can draw your molecule as well directly into this, or you can upload the SDF file as well. And then there is another tool that is called as BBP Pred, and it is generally used, or they developed it for predicting the blood-brain barrier permeability of peptides. So instead of small molecules, you can also predict the BBP permeability of peptides. So you just enter a faster sequence of a peptide, and that should not be longer than 150. So it should be between 5 and 150. And then you predict, and it will give you the probability or chances of that substance to cross that peptide to cross blood-brain barrier. Again, it is based on a logistic regression classifier based on feature representation learning that learns the most discriminative feature from existing feature descriptors in a supervised way. So this is based on supervised learning machine learning tools. And then the most used tool for ADME prediction is Swiss ADME, that is developed by Swiss Bioinformatics Institute. So here again, you can either use an SDF file as an input, or you can use Smile, or you can simply draw your structure. And the blood-brain barrier permeability prediction in this tool is based on the boiled egg algorithms, which they call boiled egg, and that calculates the blood-brain barrier permeability. And how did they develop this tool is that they took the value of human intestinal absorption from literature, and then they took the values of blood-brain barrier permeability from the literature, and then they did computation of log P, W log P, and total polar surface area. And from here, in these two graphs, you can see that the blue dots are showing the compounds which are permeable to brain, while the green dots are not. And then here you can see the red dots are the compounds which are permeable, and the brown ones are not. And then after combining these two results, they developed this model where they call it boiled egg model. And then here they conclude that this egg yolk, yellow egg yolk part, the compounds, if they fall within this yellow egg yolk part, they will be permeable to blood-brain barrier, and if they fall into this white part, they will not be permeable. So that's why they call it boiled egg method. So this is one of the most used, and I will try to show you just kind of a small demo how to use it, and what will be the limitations as well. And then using support vector machines, the Mizuguchi lab they developed a tool called TrueMap, and that I will show you. So, and then how they developed it was they used the Campbell dataset and KEGG drug dataset, and then they calculated different descriptors like CDK descriptor calculator GUI, mode rad, and petal descriptors, and then they used random forest, gradient boosting, support vector machines, radial functional kernel, linear kernel, and partial least square. So they use different methods to develop models, and then they validated those models using squared correlation coefficient and RMS root mean square error, and then they developed this tool actually. So this tool looks like this, TrueMap version 1.1, and now now I would like to show use of these tools a little bit. So how to stop sharing. Yeah, so first of all, I will just show how to use the Swiss ADME. So this is when you just type swissadme.ch into the browser, so you will end up with this. So here you take the profiles, like I just take open. So this is a kind of furosemide. Okay. So this is furosemide, which is a diuretic drug, and I click on run. So now you will see that it is running, and now you see the different results of different physicochemical properties: lipophilicity, water solubility, pharmacokinetics, drug likeliness, and medicinal chemistry. So here in pharmacokinetic, you will see blood-brain barrier permeant, and it says no. So and that is correct, because this is a diuretic drug, furosemide, and it doesn't cross blood-brain barrier, and it is already being studied. And this tool also gives us the same like similar value. And you can see that if you click on, if you hover your mouse cursor on this question mark after BBB permeant, so it says that it calculated this permeability according to the yolk of the boiled egg. So it means that this compound is falling not in the yellow part but in the white part. So that's why it is not permeating the permeable to the blood-brain barrier. So similarly, if I take another example of another drug, which is a CNS drug. So this is fluoxetine, a CNS antidepressant drug. If I run it, yeah. So then here you can see that blood-brain barrier permeant. You will see yes. So and it has predicted it quite well. So we already know that this drug is permeating blood-brain barrier because this is a CNS drug. Okay, so that is like how to use the Swiss ADME tool, the web server, Swiss ADME web server, and then I would like to show you how to use another tool that is the TrueMap. Okay, so this is TrueMap, and if you go to home, so you will see TrueMap version 1.2, drug metabolism and pharmacokinetic analysis platform, and it consists of databases for DMPK parameters and a program that can predict some DMPK parameters based on chemical structures of a novel compound. The TrueMap database contains data of DMPK parameters from curated public data and newly acquired experimental data obtained by unified conditions. It also contains predictive data using our prediction programs, and user can predict several DMPK parameters at once for novel compounds. And the platform provides a simple method to search and predict DMPK parameters and to contribute to the acceleration of new drug development. So it has following three functions, like you can search for the compounds. If you click on compound search, so you can search for the compounds, and then it will show you different parameters for different compounds. Like it collects all the most of the physicochemical parameters for these compounds. So you can just search for them. Otherwise, if you are interested in new predictions, so you just click on new prediction, and then you simply can load your SD file. We will do it for furosemide and fluoxetine. You can load another SD file, furosemide, and then you will see that there are different parameters, like you can predict the solubility, the fraction unbound in plasma, and then you can see that it has both a classification model and a regression model. So this is a 3-class classification where it will give an output in the form of like low chances, medium chances, and high chances, and then it will give a value in the form of R-square regression coefficient, and then based on regression. And then we have clearance. You can predict the clearance. You can predict the CYP metabolic probability. You can calculate the renal clearance as well. So, but we are interested in, for example, So we are only interested in FU brain fraction unbound in brain. So that will give us an idea of its chances to cross the blood-brain barrier. So then we click on calculate. So now you can see that it is calculating descriptors 8/9. So it means that what this algorithm is doing is it is based on molecular descriptors. So they have developed this model, and then this model is what this model is doing is it calculates the molecular descriptors for the input compounds, and then it fits those descriptors into that equation, and then predict the value of those activities actually. So now you can see that this compound which we entered into this server, so it gives solubility. It has high solubility, and fraction unbound in brain is 0.240, and this was fluoxetine, I believe. So you have permeability, blood-brain barrier permeability for this compound. Okay, so stop screen. Yeah, and with that, I would like to conclude this this talk. And if you have any questions, we can discuss it now. You can write your question in the chat box. Otherwise, you can always write to me on this email as well. So, and I'm also happy to host a few internships. If you are a postgraduate student or a PhD student and would like to learn some of the tools and try your hands on machine learning in drug discovery, so you're always welcome to have. You're always welcome to apply for an internship, because at IIT BSU, our training placement office, it provides an opportunity for external students to do an internship in our labs. So with that, thank you very much for listening to me, and I'm open for any questions.
Sir, we have received some question on chat box.
Yeah, so I can see there is a question that any difference between deep learning and machine learning. There is no, as I said, there is no difference. Actually, everything is like artificial intelligence, and under that artificial intelligence, machine learning comes, and under machine learning there is a subset actually. So deep learning is a smaller subset of machine learning. So every deep learning tool is a machine learning tool, but every deep machine learning tool is not a deep learning tool. So we can say something like that. So I can see there is another question that molecular docking is considered under AI. I did not get the question, but as much as I could understand, the person is trying to ask whether molecular docking is AI. So molecular docking is a tool which is being used for just predicting the binding affinity of a compound towards a protein or a receptor or an enzyme. Okay, so traditionally or classically, what has been used is people are just docking the compound into the receptor binding site or the enzyme binding pocket, and then they're doing some calculations on physics-based methods, like they're calculating the binding free energy, and then they are coming up with some scores. But now there are some artificial intelligence tools or AI tools or machine learning tools which can predict this affinity with better confidence. So definitely there is a new field emerging where people are trying to use artificial intelligence in molecular docking actually. But these two techniques are completely different. Molecular docking is a kind of computational drug design method, while AI is it's like in general algorithmic machine learning tool. Yeah, that's a wonderful question. Any free software available for docking studies? So there are several softwares available which are free and open source to use for molecular docking. So I can name a few of them. For example, there is Autodoc, Autodoc 4.2, which is being developed by the UCSF people. So and then there are different derivatives of Autodoc actually, which are people are using for molecular docking. Like Autodoc Vina is there, Samina is there, and then there is Pyrex, which is being used for molecular virtual screening, where you can um, perform docking of large number of compounds, like maybe 100,000 or 50,000 or something like that. And then there is another program called Doc, which is also open source and free, so one can use it without any license. And then there are several web servers as well, which are being used for example, protein docking, like Plus Pro is there, and then there is this Hadoc is there. So these are for protein docking, where you can dock two proteins by using these servers. There are the different servers, and then there for small molecule docking, as I said, Autodoc and Doc, and Autodoc Vina, Samina. So these are the open source docking tools which can be used definitely, and a lot of people are using that actually. Yeah, that's an interesting question actually. How much docking results are correlated with in vitro or in vivo activity of compounds? So that is a very kind of just on the basis of docking score, we cannot correlate that to the in vitro or in vivo activity, because molecular docking is something like it is just for comparison actually. So for example, if there are a lot of factors for that, because when we are performing molecular docking, so what we do is we just use protein structure, and then we have a molecules, and we try to fit that protein, that small molecule into that binding pocket of that protein. So there are different factors which may affect the scoring. For example, one factor is if we do not know where our compound is binding actually. So this protein can have different binding pockets, and if we do not know where our compound is going to bind, so that may lead to false results. Another thing is that this molecule we are using, and it has several conformations, for example, or several stereo centers, or there is a lot of flexibility in this ligand. So the pose which we got after docking, it may not be the true physiological pose of that molecule, because that in nature, in physiology, everything is dynamic in nature actually. So that is another thing. But definitely there are some tools which can be used to get the numbers which can be correlated with in vitro or in vivo data, and those tools are, for example, free energy. So recently there is a tool being developed called free energy perturbation methods. So you can use those methods, free energy perturbation. You can calculate the free energy of binding of that small molecule with that protein. But again, the most important information which we need in this case is the correct binding pose. So if if you have a, for example, crystal structure which is determined from X-ray crystallography or cryo-electron microscopy or NMR, so then you can predict it quite well. Otherwise, just for molecular docking, we cannot directly correlate that data with in vitro, in vivo data, but we can get kind of a relative. We can compare molecules. For example, if we have 50 molecules and we wanted to know out of those 50 molecules, which molecule has higher chance to interact with this specific protein. So we can dock all those 50 molecules into the binding pocket of that molecule, of that protein, and then we can rank those molecules according to the docking score, so that we will enrich our data set. We will make sure that the top ranking molecules will contain most active compounds. So that's how it is being done actually. But never believe that docking docking can replace the in vivo or in vitro studies. So these are just complementary studies, and the basic rule of computational studies is kind of garbage in, garbage out. So if you have quality data as input, then only you will get quality data as output. Otherwise, it is just kind of waste of time and resources. Sorry, I couldn't see the latest question. Yeah. So what are known data and unknown data in case of penetration and unfold of BBB? Yeah, Gautam Jena, thank you for your question. So the known data is, whenever some academic group or companies are developing drugs, so they do in vivo in vivo pharmacokinetic studies. So in animals, they inject those compounds, and then they determine the fraction of that substance that is or the concentration of that substance into the brain as compared to the concentration of that substance in the periphery. Okay, so that number is the labeled data. For example, if a compound is crossing blood-brain barrier and it has like 1:1 concentration in the brain in an animal, so that number we use and we say that that compound is blood-brain barrier permeable. Okay, so that we use and we collect all those data. So there are different data sets developed, and mostly those are from the published data. So people who work in this in vivo ADME projects, so they determine those data and they collect those data, and then that data can be used as a labeled data. So where we know that these compounds are able to cross the blood-brain barrier. So we use that as labeled data, and then we have unlabeled data, that is the test data. So where we supply simply whether we can use unknown data, where we don't know whether this compound is crossing blood-brain barrier or not, or we can have kind of a known test data as well, like where we know that these compounds are permeable, but we want to test that, and those have not been used in training that model. So that is the test set, actually external test set is that is called. Bhupinder Bhayan has a question that docking can be patented for specific drugs. So I'm not sure about patenting actually, because until and unless you do not prove that those results with in vitro or in vivo results, so it is, I believe, difficult for getting intellectual property protection on those compounds, on those data actually. Rips Bam is asking which PDB ID will be selected from data bank on what basis for any biological activity. Sorry, I couldn't understand your question properly, but as much as I could understand is you're just interested to know how to select a PDB for performing maybe molecular docking or any kind of computational studies. So for that, there are some basic rules actually. So when you open the PDB, so maybe I can just show you by sharing the screen, for example. Just a minute, just give me some seconds. Yeah, so when you so this is kind of a protein data bank, RCSB. Okay, so when you go to this protein data bank and you click on, for example, acetylcholine esterase, acetylcholine esterase, and then you can see that there are almost like 2,637 structures for acetylcholinesterase. So now the question is, which of the structures you can use for your studies, like either computational modeling or whatever you are going to do, molecular dynamics with that with those structures. So the most important criteria for selecting it is the species actually, like because you are interested only in humans, so you just click on Homo sapiens, because then you will see that there are 834 structures which belongs to human, or the structures which are human acetylcholinesterase enzyme. Okay, so that is one thing. Another thing is the resolution. So you can see here is the refinement resolution. So there are this is the another important aspect, because whenever these crystal structures are solved by using X-ray diffraction methods. So X-ray diffraction the higher the number of resolution, like if it is 4.5. So at this resolution, you will not be able to see the atoms properly. Okay, so that should be, I will say, like lower is better. Like zero point 0.5 to 1 is the best. 1 is the best, where you can see every single nuclei, every single atom. But I think if that is not possible, then you can go for one below less than 1.5. Okay, so for example, if you search that, and then you are having 50 structures, and then is so this is like, so for example, if you, or maybe I will just keep it the highest resolution, like this one, and then you see that there is only one structure. And if you will look at this, so you can see the electron density here as well. So you see the electron density, click on electron density, and then you will see how the electron density looks like, so or how it looks like when you collect the X-ray diffraction data. It's now showing some problem actually. Yeah, but as I said, the most important two most important things are, one is the species which you are using, and another one is the yeah. Now you can see the electron density. So here, for example, if you look at the electron density, yeah. So now if you zoom it a little bit, so here, for example, this side chain, you can clearly see each and every atom actually. Okay, while if I compare this with, so for example, this one. So you can clearly see the electron density, and you can clearly see the atoms. But if you compare it with a structure which is Yeah, so you can yourself compare it actually. So you can compare it with higher, low and high resolution structures. And another important thing is R value. So that is when you, because what you are doing is you are fitting that the observed data into the into the model, your model. So the R value should be close to zero actually. So the lower the R value, uh, better is the model. So these three things, I think if you are keeping these three things in mind for choosing a PDB structure, that will be the best. You will get a best PDB structure. Like the resolution, lower the number, higher the resolution, lower the R value, better is the model and the species. Yeah. Any other questions?
Thank you, sir, for your nice presentation. You have correlated all related to artificial intelligence and also computational approaches, how it can be applied for drug design and development. So all these things you have covered under this presentation. So we are very much thankful to you, sir, for your nice presentation.
Yeah, thank you so much, Professor Sahu. Thank you so much for providing me the opportunity, and I'm very happy for this interactive session.
Thank you so much, Dr. Rajni sir, for sharing your vast research experience in neuroscience with all of us. Thank you for making time. Thank you for making time to sit through this presentation. This presentation was very lively, engaging, educative, and fulfilling because of your presence, sir. All our participants are going to be benefited from your breadth of knowledge and depth of expertise. May I now request Principal Professor Dr. B.V.V. Ravi Kumar, sir, coordinator of this event, to present our venerated resource person, Dr. Rajni, sir, with an e-memento as an expression of our gratitude and its inexplicable bond we have forged with him.
Thank you, Dr. Rajneesh, for sharing your valuable time and giving wonderful information. And I'm sure that it will be definitely a great help for the teachers who are working in this area of research. Thank you once again for accepting the invitation. On behalf of Roland Institute of Pharmaceutical Sciences, I would like to present a certificate of appreciation kindly accepted.
Yeah, thank you so much, sir. Thank you so much.
Thank you, sir.
Thank you, sir, for sparing your valuable time for us. Dear participants, with this, the session 1 of Day 5 Series 2 FDP comes to an end. We will be resuming back the session 2 of today's event at 2 p.m. on the same platform. Thank you all for your active participation and kind attention.